Hi, my name is Karen Gans, and I'm presenting this little video because I'm an alumni of the SynBio mini course that was put on by Nathan. And so I'm just going to give you a little overview of the course and then how we implemented the material here at the university that I teach at. So I'm doing this with a crutch. I'm going to use my PowerPoint to sort of do this, give me some highlights and help direct my talk. So let me share my screen really quick. And here we go. So, so again, my name's Karen, and we are using the SynBio methods and background at New Mexico Tech. New Mexico Tech is a small institution. It's a small public institution in New Mexico. We are about 1,300 undergraduate students, so we're small. We also have graduate division, so we're not just undergraduate serving, and we also have research. So we're very big on research oriented. We've always tried to get undergraduates involved in research, and that's been a big draw for students coming to the university. I am in the biology department. My role here, I was hired as the lab manager, so I'm in charge of developing the labs for the students, overseeing the TAs who teach the labs. But I've sort of, over the past couple of years, evolved into teaching some of the undergraduate courses and also teaching in some other courses here at Tech for teachers. We also have a program for getting teachers trained so they get their certificate, and also we have a Master's of Science for teaching, which is a grad program to get teachers their master's degree. So I'm involved in a lot of teaching. I have a background in that, and we were looking to implement some of the course-based undergraduate research experience courses here at Tech, and I heard about Nathan's class, so I signed up to participate in this class in the summer of 2017. So in the summer of 2017, another professor and I from Tech were supposed to come out for the summer to do this 4-day course, which I thought was just going to be a workshop on the course-based undergraduate research experience. I didn't realize that it was going to be focusing on the synthetic biology as much as it was. So I kind of went into it not really understanding at all what I was getting into, and I had never been to Arkansas before, so that was really interesting. And I really appreciate the hospitality, though, of Nathan and Raina and everybody that was involved in that. We had a great time, and it was. I'm sorry that you don't get to visit the campus, but hopefully you can stop in at some point. So summer of 2017. That's now whatever 3 years ago that we went, or I went. The other professor actually wasn't able to go, so I was by myself to do this. So I had a great time, from the get-go. Immediately Nathan immersed us into what the techniques were for doing this, and that's exactly what you want your students to be doing. Just, you get immersed. There's a little bit of background, but the hands-on is where you learn, and that was immediately evident, and all the pieces sort of come together. You get little bits and pieces, but then it all comes together, and it all just worked. So we did this workshop, got involved, and understood what the iGEM system, or the International Genetic Engineering Machine, which was the background for the synthetic biology course, was this whole program that was developed at MIT. And so through the program, we got involved in the iGEM program, and the workshop paid for the $500 fee for the lab fee to be able to get the parts from the iGEM system at MIT. So leaving the workshop with all these tools and then the availability to have these parts to be able to do the synthetic biology stuff at Tech, we left the workshop. I wasn't able to start implementing things until the summer of 2018, where I did a pilot class for my Master's of Science for Teachers course. So this was a 2-week intensive for teachers. And I involved them with the parts, and I'm teaching teachers who don't necessarily have a background in biology. Well, they have a little bit of background, but they're certainly not in the lab. And the system was so straightforward that I was able to run this pilot course and sort of get things up and going. So then what happened was the next course that I implemented on this was in the fall of 2018. We have a program at Tech called the Biomedical Sciences Program, and this is a hybrid engineering science degree program at Tech. And we had been implementing and giving students more hands-on research experience as part of the curriculum. And so this course fell right into that. This is geared towards sophomores, so they've only had 1 year of biology at this point, and it's hands-on. I run it so that once a week I have a group powwow. I give them some background into the techniques, and I give them a task of what they're supposed to do that week in the lab. And then they have to do that assignment during the week. And again, we're basing it off of the iGEM system and introducing restriction digest, cloning, ligation, transformation, PCR, all of those techniques. Every week I sort of cover those techniques, and then the students go into the lab and do the course. So it is now part of our curriculum. It's required for the BMS students to take this course. And so ever since then, I've been doing this sort of alternating in the summer, doing this course for the teachers during the summer and then the students in the fall semester. This is just an ad for the first time I ran this course, and it's a 3-credit course. The first time I ran it, of course, the students had to meet at one particular time. We chose Friday from 2 to 5, and that's when I was giving students this lecture, and then they would have to come in on their own during the course of the week. So we were still trying to figure out how to give students credit and the time requirement that for the expectation for them to come into the lab. So that was the first semester. Second semester went a lot smoother. And then the third semester, well, we'll see, because the third semester is 2020. So I just want to tell you once you start doing these courses, a whole bunch of other stuff just starts opening up. So this is the course listing on the left-hand side. Our course is BMS Biomedical Sciences 301. I call it Genetic Engineering at the Bench. It's now a 2-credit course, which is a 1-hour contact and then 3-hour lab during the week. And again, they come in, we have an open lab for them to be able to do that. We also developed another CURE course as part of the BMS program, and this is a senior level core class. So it's also required. So they take the sophomore course, which is more directed, and then the senior course, then they're ready for much more independent research. It's also course-based, and at this point, it's run by one of the researchers at the university. So they lead the class with the research that they are conducting in their laboratory, and students partake in that. And we've even expanded even further. Last year, we applied to the CPHAGES program, which is out of the Howard Hughes directive. And this is a well-known national CURE class. But because of our background and because of our setup now at the university, we got accepted into the 13th cohort for CPHAGES, which we were all ready to start for our freshman class in the fall of 2020. Unfortunately, we had to delay it a year because of the coronavirus. But you can see how once you start getting these CURE classes sort of implemented and you start seeing how the students respond, then you can start building the curriculum and students get different levels of experience and are able to have it throughout their career at the college. So lab space, I've sort of touched on it, was really key for being able to implement this class. We dedicated a whole lab now to these CURE classes. That means that it is open pretty much, I don't want to say 24/7 because we don't allow our undergraduates in at night, but it's open during the day. So during the 8 hours during the class time, students are allowed to come in. It's stocked with what they need, the equipment that they need to be able to work on their own, you know, freezers, refrigerators, incubators. We just got an autoclave that we put in there that's very student friendly so that they can actually autoclave their own stuff, pour their own plates. My job is lab manager here at New Mexico Tech, so you kind of need someone like me around to be able to step in if a student needs help. So I'm here most of the time, and that's sort of a key part to being able to implement these classes. And so to take this even further, when we have a program here called the INBRE program, which is out of NIH, comes from the Idea Networks of Biomedical Research Excellence. INBRE is sort of this, it's a statewide program that pulls all the universities together, and we share biomedical research, and it's up and coming way for researchers to get money and for education to be implemented at the other universities. Our new funding, our 5-year funding, happened 2 years ago, and they approached us to sort of drive the theme of our next 5 years, and the theme is called Experiential Learning Development Program, and it's based on CURE classes because of the success that we had with the CURE classes here from the SynBio. So our 5-year INBRE grant is now focused around the universities developing these CURE classes at the separate campuses, and of course we're using that to develop them more here, but I'm also going out to the other universities and letting them know how to start developing a CURE class at their campus. So yeah, it just kind of blossomed. And not really sure what's gonna happen in the future. This year, this whole coronavirus has sort of put a damper on stuff because of not being, we're not sure if the university is going to be open. We're not sure if it's going to be distance. We're not sure what the program is going to be. And of course, these CURE classes are so hands-on, it's difficult to be able to see how we might do teaching completely online, but we're going to embrace it. iGEM is embracing it. They are doing, they did a virtual activity for their 2020 festival year. And I just want to say, actually, one other outcome of this is one of the students has decided that they want to develop an iGEM team here. So next year, they're going to do the fundraising for the $5,000 to become a team member as opposed to the lab component of the iGEM. And so anyway, so everyone's going virtual. Everyone's trying to figure this out. Nathan and everybody at Arkansas is trying to figure this out. That's why you're part of this virtual workshop, which is fantastic. And we're just going to keep it going because it is incredibly successful, and it just people hop on the bandwagon, and students love it, and it just provides them the confidence that they need to go into their other lab classes and to go on with their careers wherever they're gonna lead them. So have fun, enjoy, and thank you everybody at Arkansas for starting me on this path. It was life changing.